Hello, everyone. In my presentation, I'll talk about how drug polymer complexation between a high pK weakly basic drug and an ionizable acidic polymer HPMCAS impact dissolution of amorphous solid dispersions. Amorphous solid dispersions are a widely used strategy for improving solubility and dissolution rate of poorly water-soluble drugs. Upon dissolution under non-sink conditions, ASDs ideally form a supersaturated drug solution that leads to increased membrane transport and bioavailability in comparison to the crystalline drug. ASDs primarily consist of a hydrophobic drug molecularly dispersed in the matrix of a hydrophilic polymer. HPMCAS is the second most used polymer after PVPVA in formulation of ASDs. It is an ionizable pH-sensitive polymer with an acidic pK of 4.97. Solubilization of HPMCAS occurs in 4 stages. First stage is diffusion of water molecules into the polymer matrix. Second stage is ionization at a favorable pH with liberation of protons. Third stage is hydration, which happens only when polymer is sufficiently ionized. Fourth stage is diffusion and solubilization into the bulk solution. Because of ionization and liberation of protons, gel layer pH could be less than the bulk solution depending upon the buffer capacity of the medium. Therefore, polymer ionization, its interactions with the aqueous environment, and drug polymer interactions can considerably alter the dissolution outcome of HPMCAS ASDs. The objective of the current research was to investigate drug polymer complexation between a weakly basic cationic molecule, bedaquiline, and an ionizable acidic polymer, HPMCAS. The second objective of the research was to develop a biorelevant dissolution method that can overcome drug polymer complexation. Bedaquiline is a weakly basic molecule with a pK of 8.51. It is practically insoluble in water and has a high log P of 7. It has therefore been classified as a BCS class 2 molecule. However, it has twofold bioavailability when it is administered with food. Bedaquiline has a crystalline solubility of around 60 microgram per ml at pH 1.6, which drastically reduces with increase in pH to 6.5. However, we observed a dramatic improvement in crystalline solubility of bedaquiline in biorelevant media. In fasted state version 1 medium, bedaquiline has a solubility of around 7 microgram per ml. In fed state version 1 and version 2 media, bedaquiline has a crystalline solubility of around 600 and 250 microgram per ml respectively. At pH 3, bedaquiline has an amorphous solubility of around 45 microgram per ml, which drops down to just 0.6 microgram per ml at pH 6.5. We prepared ASDs of bedaquiline and HPMCAS using solvent evaporation method. 5, 10, 20, and 30% drug loading ASDs were confirmed to be amorphous from modulated DSC and PXRD analysis. We then performed two-stage dissolution of 5% ASD under non-sink conditions with a target dose concentration of 100 microgram per ml. The initial pH of the dissolution medium was 1.6, and at 60 minutes, we used a concentrated buffer to switch the pH to 6.5. At pH 1.6, bedaquiline release was only 10%. This is because HPMCAS does not ionize or solubilize at this pH. With switch in pH to 6.5, we still only observed a slight improvement in bedaquiline release to up to 25 to 30%. And at the end of dissolution experiment, we observed this insoluble residue at the bottom of the dissolution vessel. Upon PXRD analysis, we confirmed that this residue was rather amorphous. This clearly ruled out matrix or solution recrystallization of bedaquiline as the probable cause for poor release behavior at pH 6.5. Moreover, from FTIR analysis, we confirmed that this residue consisted of both HPMCAS and bedaquiline. We then investigated drug polymer interactions and possibility of complexation between bedaquiline and HPMCAS during dissolution. Drug polymer complexation was investigated using a co-precipitation method. For the study, we prepared two sets of samples. In the control samples, we dissolved HPMCAS stock solutions in buffer pH 5.8, 6.5, biorelevant media, and pH 6 buffer, which corresponds to the gel layer pH. In each of these media, we obtained a clear solution, and after ultracentrifugation, we analyzed each of these media for polymer concentrations using UV visible colorimetric method or HPLC equipped with ELST detector. In the test samples, we dissolved bedaquiline and HPMC AS stock solutions in the same media. However, in this case, we observed turbidity and precipitation. This is because both bedaquiline and polymer are ionized under these pH conditions and consisted of two oppositely charged functional groups. Because of electrostatic interactions, they formed an insoluble hydrophobic ion pair complex. We separated this insoluble complex by ultracentrifugation, dried it overnight, and analyzed it in FTIR. The supernatant were analyzed for polymer concentrations. We use this equation to calculate percentage polymer complexation in each of these media. The carbonyl peak observed at around 1720 confirmed presence of HPMCAS in the insoluble complexation residue. Presence of bedaquiline in the insoluble complexation residue was confirmed from aromatic CN and aromatic CC peaks observed at around 1250 and 1450 respectively. This next image here depicts percentage polymer complexation when stock solutions containing different molar ratios of bedaquiline to fixed number of moles of carboxylic acid groups of polymer were added into different media. Each of these points when vertically red represents 5, 10, 20, and 30% drug loading. We observed 90% polymer complexation at pH 5.8 and 6. When pH was 6.5, percentage polymer complexation was 60% and lower. This was because of greater extent of polymer ionization, hydration, and solubilization, which reduces its complexation tendency. In biorelevant media, percentage polymer complexation dropped down to just 20%. This is because of micellar solubilization of bedaquiline. Drug polymer complexation was disrupted, which freed carboxylic acid groups of polymer for hydration and solubilization. These complexation results helped us in explaining poor release behavior at pH 6.5, and we also estimated that release behavior of bedaquiline HPMC ASTs would be considerably better in biorelevant media. At pH 6.5, we observed 30% bedaquiline release from 5% drug loading ASTs. When drug loading was 10%, bedaquiline release dropped down to 10%. This is because of drug polymer complexation, which prevents polymer hydration, solubilization, and release of bedaquiline from the polymer matrix. In fasted state version 1 media, which also has a pH of 6.5, however, it additionally contains solubilizing micellar species, we observed complete bedaquiline release from 10% drug loading AST. When drug loading was increased to 20%, bedaquiline release dropped down to 80%. However, release behavior in fasted state version 1 media proved disruption of drug polymer complexation in presence of micelles. In fed state version 2 media, we observed complete release from 20 and 30% drug loading ASTs. This is because of fairly high crystalline solubility of bedaquiline in fed state version 2 media, which is 258 microgram per ml, which is why the dissolution conditions in this experiment were no longer non-sync. The conclusions drawn from the current research work were that HPMC-AS has the tendency to form hydrophobic ion pair complex with weakly basic cationic molecules if they are ionized at a particular pH and consist of two oppositely charged functional groups. Drug polymer complexation can impede dissolution because it prevents polymer hydration, solubilization, and release of drug from the polymer matrix. With increase in pH, percentage polymer complexation reduces because of greater extent of polymer ionization, hydration, and solubilization. Micelles, on the other hand, disrupted ion pair complexation by solubilizing bedaquiline, which freed carboxylic acid groups for hydration and solubilization, which is why we observed improved release profiles in fasted state version 1 and fed state version 2 media. However, it is important to establish relationship of in vitro biorelevant dissolution testing with in vivo performance of such complicated systems. Thank you.